speaker for this afternoon is not really new. In fact, I'm very happy to say she's one of the former LSU students. She did PhD work here in 2006, and now she's one with us. She's a scientist. I didn't also know that we have a position as associate scientist. I thought it should be just, scientist, but well, I have to say that, so she's a scientist with us, and she's working with a group of Dr. Vincent, and she's a crop physiologist. So without much ado, let me call on Dr. Jana Kholova. And she's also very Indian, I have to say, because she's married to an Indian guy. So ladies and gentlemen,
and moreover, about the techniques which we think can actually contribute to acceleration of breeding strategies. What is not only, in my opinion, the burning issue at ICRISAT, but also worldwide. So let me start like this. Whether we like it or not, the crop yield and crop production in the field is still big black box. Even after so many years of scientific effort, this crop production in the field is so complex trait that it's hard to understand. And therefore, probably that is the reason why we cannot progress or accelerate the yield improvement. So the way how I anticipated we can progress with the issue of yield improvement is to dissect and understand the interaction between the crop, like our genotype, environment, and management. And from this regard, crop model tool is the excellent tool to dissect and understand these interactions. So for the purposes of this presentation, I will just keep aside the management improvement strategy, and I will talk about what do we do regarding improvement of our understanding and applicability of our understanding of interaction between genotype and environment. So this messy slide should show what is our approach. First, even before starting talking about the crop, we should probably make clear and few clear questions we should ask ourselves. So the first one is for which regions we actually want to breed, where our crop improved variety has to perform. In this case, this is the 3 states of India, Andhra Pradesh, Maharashtra, and Karnataka, and this represents the highest productive regions where the rabi sorghum is mostly grown. So this area has a lot of characteristics, but definitely it is not uniform. So first of our exercises was to partition this whole area in a little more homogeneous places. So what is highlighted here, for example, in red light or in yellow borders. So now it comes for our first modeling exercise. We should ask what would be the major constraint for crop production across this area. So here we can employ our model, in this case APSIM, to answer some of these questions. So here, when we use APSIM and grow Maldandi genotype of sorghum, which is the most common rabi sorghum cultivar, we obviously found out that there are huge restrictions in water, huge water limitation on crop production. How I will just show you here on the x-axis, follow with me, there is the crop cycle, and here on y-axis, there is a measure of stress. Which is in the form of water supply-demand ratio. So as the supply-demand ratio declines, the crop suffers more from drought. So we have characterized each of the cropping season across this area, and we were able to cluster these crop trajectories or environments which crop is facing with regards to the water availability across the season. So you see that there are various types of stresses with terminal progressions, but there are also quite plenty of seasons where crop is not facing much of the water limitations, and also some type of intermittent drought stresses. So then we have to ask, if we have these limitations, what does it mean for the crop production across this area? That is shown on this graph. You see that the stress which comes either before flowering or at flowering nearly completely devastates the crop yield. However, as we move to the milder stress scenarios, there is not much of negative effect on the, at least on the grain yield production. I have to look at you guys also. Okay, so after all, it's not modeling but modeling.
Like this.
Fine. So now, I hope you, in between, you anticipated what I meant with this slide. So this is the yield devastation, and only after we are a little bit clear what our crop is facing, what is its limitation, we can start asking about the crop's features, which actually can confer the advantage in the most frequent of all these environments. And frequency is what really matters here. Because, yes, and for our case, this is the frequencies of all those drought stress patterns for our area of interest. You see that nearly, or in 2 out of 3 seasons, our crop faces certain water limitations. And you have seen in my previous slide that in these 2 scenarios, which are really quite frequent, in our Rabi sorghum tract, the yield is nearly devastated. So let me go to the next slide. This was another exercise which we did with the use of an EPSIM model. Here we have tried to characterize what is the best characteristics which determines the yield in our area of interest. So we found that it is actually the amount of water which remains in the soil profile after anthesis, which is the best factor which can explain our yield variability. So in this graph you see that this yellow spots, that is yellow and brown spots, those are those most severe stress scenarios. And in these most severe stress scenarios, the yield is vastly and very tightly determined by the post-anthesis water use. However, as we go farther and as we increase the water available in our environment after flowering, you see that the correlation becomes little loose. So there are probably other factors which determine and influence yield, and which will be more important for the crop in this situation than is water. So now let's finally talk about the crop. Now we are a little clear what is our environment. Let's talk what do we do in a research regarding our crop and regarding the improvement of the genotypes. So from this point of view, we got amazing populations or germplasm from our breeding group, which has developed 2 populations of sorghum, which are based on the senescent sorghum R16 and S35, which has been introgressed with the various QTLs which were proved to contribute to the stay green. But again, it is not stay green as stay green here itself. I should make sure what do we mean in our presentation by stay green. So in our case, I mean the stay green phenotype is such a crop which is able to maintain its green leaf area for longer time after imposition of terminal drought. So from this regards, we analyzed our populations and we found huge variability for hydraulic conductivity of plant tissues within these sets of introgression lines. I should denote here that this is the constitutive trait which actually we are able to observe in a well-watered condition. So another type of traits which we observe is canopy size, and there is substantial variability for canopy size in our set of stay green isogenic lines, and not only canopy size, but also certain parameters which relate to the canopy developmental dynamics. Again, it is constitutive trait which we can measure and see under well-watered conditions. And finally, we were also interested and found certain variation in traits which determines plant growth under water stress. So let me move what we found with regards to the hydraulic conductivity of plant tissues and how we were able to reproduce the same variability with models. So on this slide, in the first part, we see the research indication and the previous research before we even start working with sorghum. So all these mechanisms related to hydraulic conductance of the plant tissues were originally explored in pearl millet. So I won't make it too complicated and complex for this presentation, but we were actually able to identify the mechanisms which relate to midday stomatal closure. So in pearl millet, we found such lines which were able to transpire here on, which were able to transpire proportionally to the increasing vapor pressure deficit. What I mean is that these lines were able to transpire on maximum even if the air during the midday was really hot and dry. But we had even such genotypes which were able to restrict their stomata conductivity if they were in the environment where it was really hot and dry. So we found that this variability in canopy conductance is probably also somehow linked to the differences in hydraulic limitation in the root tissues. So this trait is actually very, very smart and very broadly useful, and we have identified the variability for this trait not only in millet but even in the populations of another mandate crops and of course even in our sorghum. So this is the variability within our sorghum stay green isolines. So upstream provides us with certain coefficients which can reproduce and reflect these real observed traits. So in one way, we can mimic this mechanism while imposing the limitation on maximum transpiration in Epsim model. In another way, we can get close to the simulation of this phenomenon when we limit the root hydraulic conductivity with the KL constant in Epsim. So I will just show you example of what happens when we introduce this trait to the Maldandi original sorghum genotype. So that you may agree with me that looks quite reasonable. Here on x-axis you have various stress scenarios. On y-axis you have proportional increase in yield after introduction of this trait of hydraulic limitations in crop canopy to that original genotype. So you see that across various zones in our main Rabi sorghum tract, we are getting quite reasonable yield increase across all those various scenarios and all the regions. So here this graph shows the same. Again here on x-axis, follow with me, is the original yield of original Maldandi virtual crop simulation. And here on y-axis, you have either yield benefit, which is positive value, or yield decrease due to the introduction of that trait. So if we add this trait to, or if we virtually introgress this trait to M35, to that common Maldandi sorghum, you see that there is a flat increase in the amount of grain yield across all those scenarios of stress patterns. And moreover, because we should not think in crop production only in the sense of grain yield, also what is important on sorghum is stover, and therefore we need to, if our improved genotype has to be adopted, we need to produce such which would not sacrifice the stover production. So in this case, you see that this trait of hydraulic limited conductivity has actually positive effect even on stover accumulation. So if we go further, now I will talk a little bit about our variability which we identified for canopy size. The real observations are showed here that S35 is senescent parent and 7001 is one of those successful introgression lines. So here you see that 7001 actually produced less leaf area, and this variability was really very strict across the year. So again, in UpSim, we have the provision how to mimic this mechanism. And if we introduce this trait to M35, or first I will show you on this, as shown on this slide, we have a really good research indication that this leaf area at antithesis, means maximum leaf area attained by the genotype, actually links very well to the grain yield production after in a very severe stress scenario. And these data are from our lysimetric system. So now let's go for let's see what happened when we did the simulation of this trait across the rabi sorghum tract. So here you can see that there is actually very different effect of this trait in various zones of our rabi sorghum tract. So the most beneficial across this or according to this graph, this trait would be in that production core of rabi sorghum tract. So again, if I show you the same, only visualized in a different way, you see that the worst stress scenarios would benefit, or in the worst stress scenario, our crop production will benefit from introduction of this trait. But there will be definitely trade-off with the stover yield quantities. Another question is, which we are currently not able to answer with the existing models, what would be the effect of this trait on stover quality or on the grain quality, which is very important to know if we want our technology to be adopted. So currently we are working on developing the loops in UpSim to actually reflect the quality of stover or yield, which can further give the value of each of the technology which we are developing nowadays. So again, this is just for comparison. This is another characteristics of canopy development, which I try to approximate through models. And for the first look, it looks the same. However, you follow with me the red genotype is again our senescent genotype R16, and K369 is one of the best introgression line on this background. You see that these lines has attains quite similar maximum leaf area. However, the way how do they reach this point is very different during the crop cycle. So we were interested to mimic this canopy developmental dynamics and to see what will happen with the yield of such a line which would possess this mechanism. So again, in UpSim, we can reflect this variability using various coefficients. And this is what happens after we improve or after we virtually introgress this trait to our sorghum. So you see that again, there would be the best proportional benefits of this strategy in terms of grain yield in the most severe scenarios which exist in our area of interest. And within those, it would be beneficial across all of those subregions or subzones which we identified within major rabi sorghum tract. So again, I will try to explain more on this graph. So again, this is the same. You can see that with regards to grain yield, there are certain improvements in the grain which is symbolized in those yellow and brown spots, which are actually the most severe stress scenarios. However, if we introduce this mechanism, we would cut flat on the production of stover. So again, we need to see each mechanism from both sides. Each coin has two sides, no? We need to evaluate what would be the grain yield, but we have to again see how adaptable or adoptable the strategy will be from all various angles. So again, there is the question if that stover yield trade-off would be compensated by the improved quality or not, but that is the question for our future research. So now let's move to the last sort of traits which we investigated. I was talking, those previous traits were constitutive, they were the constitutive features of the plants which we could observe in well-watered conditions. Now let's move to the stress response of the crop. So even with this regards, we found quite huge variability in our isolines. So you try, this is again a little bit complicated to follow, but you try with me. Here on the x-axis, we have progressing terminal drought. Here on the y-axis, we have increasing leaf area. And the same colors represent the same genotype, and full dots are the control conditions, and empty dots is the same genotype under water stress treatment. And in this case, it's the early water stress which was imposed long before flowering. So we have here two strategies. Apart of that, for the first look, you can see that our genotypes are very different in maximum leaf area they attained. And then we have another sort of variability. You see the first one is this red genotype, which is S35. It's our senescent background or senescent parent, which actually develops very similar amount of leaf area regardless of water treatment. The similar things you can see even in this pink genotype. However, in other lines, we found that they are actually able to restrict the leaf expansion when they sense the drought. So again, upstream provides us several ways how we can reflect this variability in a virtual crop. And now the quiz, if you are sleeping or not. What do you think, what will happen if I prepare the virtual in silico genotype which will contain or which will be in progress with this feature? Will it increase yield or will it decrease yield in very severe stress scenarios? You just get something. Increase. Fine, you owe me coffee. Yeah, I was surprised myself because I would think, okay, the best of the lines of stay green actually contain this trait, so it should be some beneficial one. But when I tried to manipulate with upstream to reproduce the same variability, it actually led to the yield decrease. What was really interesting. And that is what is the magic of the upstream, that it will account to various, the full environmental complexity which in our insight we are not able to capture. So again, I will visualize the same. It definitely looks like a trait which we wouldn't want, which wouldn't be desirable in our conditions of whatever, of Indian subcontinent. Yes. So again, there would be the trade-off. As I showed, as you have seen, the decrease in a grain yield would be actually compensated by the increase in a stover gain. But still, it is quite surprising fact. So we have to still ask, because each crop model is just a tool, and it has been developed by the people. So obvious question is it really working in the right way? So that is still what we need to investigate. Or is the trait which we observe, the limitation in leaf expansion, just a consequence of other mechanism which is finally beneficial for the crop? That is what will, so this is also one way how we can use the model to to actually raise another question for further research. So now, till now, I was just talking about the crop as in a very static way. I have some input, I have some output in terms of stover or grain yield, but what would be actually the driver of the yield advantage or loss? So what is the driver? I am trying to show you on this slide. It is a little complicated to understand, but you follow with me. So in this slide, I have visualized both of the genotypes, one which has the mechanism of limited conductance or limited stomata conductance, or in another way I can still say it has midday stomata closure mechanism, the blue one, and the red one doesn't. So what is happening during the season? This graph represents the only one season in Solapur, what is the main core production of our rabi sorghum. So here, as the season progresses, we see our crops are growing very similarly. Up to the point, they are growing leaf area, they are growing biomass, and actually because this is one of the most severe terminal drought stress scenario, there is limitations on water. There is only some water in the profile and there is no rain in the season. So our crop progressively exhausts its water resources. And you can see it's happening really in a different way in the blue genotype compared to the red genotype. And here the red genotype actually exhausts its water even during the vegetative growth, and then it crashes far earlier or little earlier compared to the blue genotype. And moreover, this slight water saving in a vegetative growth of these different crops actually leads to the delayed senescence. It is really amazing because we can simulate, this means that we can simulate and reproduce the stay green phenotype with differences in midday stomata closure. That's already, I feel, a great result. And moreover, these slight differences, sometimes the differences in water extraction after flowering are really very minor. These small amounts of water saved in the soil profile actually leads to the complete crash and yield failure of the red genotype. However, the blue one is able to produce at least some yield, some 300 kgs per hectare. So this is only example, but in the case of other mechanisms which I showed, like those canopy development or responsiveness of the crop to the drought, actually the consequences of these mechanisms were very similar. Always any yield gain revolved around the conservation of soil water during the vegetative growth of the crop. And I was, at the beginning, I was telling you that I want to talk about the implications for breeding. So here it is. So our ultimate effort is to scale up and convince breeders or guide the breeders' decision if they want to use the surrogate traits instead of yield, which should be maybe more accurate. So one of our big aims for the future is to provide actually the landscapes across huge areas of the crop growing areas, and to provide some tool for considering, okay, if you develop crops with such traits, it can be really far more useful for the areas, for example, in this case, which are reddish, because the probability in yield increase is higher in these areas than, for example, those which are yellowish here. Or in the case of India, I'm for time being able to do it only in a case of full regions. So you see that, for example, for these states which are districts which belong to the sorghum production core, certain traits may contribute to the yield advantage with far better probability than, for example, here in Anantapur. So that's our ultimate effort. And obviously this is not only my work, only work of our team of physiologists to do all these complex exercises and to understand the crop production and its possibilities for improvement from various angles. We actually need to engage many teams and experiences from many sites across the globe, and we are including many people from various places which has very good experience in physiology. We obviously have to collaborate with breeders, and of course our research group contains also involves many modelers. So this is it. Thank you. I hope you are now clear about stay green in sorghum.
You have any question? Especially with our young researchers. Yes. Yes. Push.
So you showed like all the traits only for drought areas, like under drought conditions, how they are performing. So that's what like each, for example, the VPD trait and all. So did you work on that, how they are like under well irrigated conditions and low drought conditions? So
yes.
Yeah,
actually, I have done a shortcut because that would be for another lecture to just explain where is our research currently on these particular mechanisms. Like you know yourself because you work on this very well that this trait is not with the limitations. I am just showing very simplified version of what we understood and how we can model it, because current models we cannot really encompass all the complexity of the plant behavior as per now. But our further research should actually well identify the limitations of model and try to introduce our experience of this trait behavior, because currently it's very static. We just cut flat in model on a transpiration. Obviously it is not what plants are doing. No, so with the progressing research and with the very rigid proofs of the behavior or dynamics of these traits in real plants, we should be able further also to improve even models. So I showed a little bit on what would be the plant behavior if the trait is under mild water conditions in terms of yield and stover, no?
Yeah, I understand that. For example, if you see that VPD trait, like response of the transpiration under various vapor pressure deficit conditions, based on your model in sorghum, it shows that yield increase. So, but basically, if you see soybean and all, it shows like yield penalty.
Yeah.
So, how come like it's showing yield increase? Because it will have like penalty. Like if it is restricting its transpiration, means it should restrict it photosynthesis also and carbon
Thanks
accumulation.
a lot for this question. I hope I will not spend half an hour for answering it. Yes, actually, this is very good. In the model, we have currently two ways how to do, and even in science, it's not clear if the limitations are of this trait expression is caused by the hydraulic limitation of the leaf or hydraulic limitation of the roots. So in this case, I have just shown the amazing case of modeling hydraulic limitations of the leaf. But in the case I impose the same hydraulic limitations in the roots, I will get exactly what you said. In stress scenarios, I will have yield benefits. But in the well-watered conditions, I will definitely have a yield penalty in case of stover and in case of grain yield. If I impose the limits on the roots at the side of the roots, because obviously at the leaf level it won't affect the plant growth much as per the model, as per the current model. But if I limit the hydraulics of the roots, then even model is responsive enough that it affects like all the growth processes. So as I like to say, the middle way would be probably the best. So even I have done some exercises which will club these two things together. I can create even the crop which will have hydraulic limitation at the roots and at the same size at the leaf level. Like we cannot do it, for example, for the publication because we don't have enough of evidence from the research. But if I have done this theoretical exercise, we would actually have exactly what you have said, and which would correspond to the best to the things we see in the crop.
And it's not one more question. What could be the reason behind that limitation on the leaf expansion under drought condition resulted like a low yield under drought conditions?
Yeah.
So
Thanks a lot.
technically you should have like increased the yield. Do you have any idea?
I don't have any idea as for now. I feel that the crop model is actually not reflecting all the complexity, or I have chosen the wrong coefficient to reflect it.
Is that because like it will have like during the grain development the leaves will act as source, like source, isn't it? So because they will have less source, that's why the yield
Yeah, you
has
probably
dropped.
hit the target. I would say the same. That despite we would think that if they restrict their leaf area, they should utilize little less water, which can be used as for the grain filling. But actually, these virtual crops, they do not do that. They just restrict their leaf area growth, and then probably they just sit there. I am yet to produce exactly this exercise.
So because they will have less source at the time of the grain development.
Yeah, I feel it's probably for that reason.
Thanks, Jana. Fascinating seminar. I was wondering, so stay green, there's multiple QTLs. When you do your virtual introgression, do you do one at a time, or you introgressed all of them, or?
Actually, I do not virtually introgress the QTL, but I introgress the mechanism. We are, as physiologists, we are not bothering much about the genetics. We just bother about the phenotypic variation. And once we know which phenotypic variation we want, we tell breeders, okay, you make me the crop which will have this, or introgress the stay green, or whatever part of the chromosome, and this should be your target phenotype. I hope no one will shoot me from my team now.
So in this case, which phenotypic variation did you take, for one QTL or for all of them, or what was the phenotypic variation?
Yeah, actually that introgression of QTLs was one by one, and in some rare cases they have developed even two introgressions coupled in one genotype. But the truth is that there were certain inclination that particular, that would be again for another seminar, that one QTL could cause certain variation in the phenotype in one background, but it could have exactly opposite effect in another background. Even such variation we had. So it's I better don't like talking about the stay green like one trait or one part of the genome. I don't know if it is answering your question.
For me, I'm social science, but I really like to point out one thing. It's sort of like testing the question with one partner. You mimic what you wanted to do, or you simulate what you wanted to do. What's the performance of the technology? That's the question all about simulation, right? That's my question. To what extent are you going to simulate each time? Because you have several factors so that you make the decision what should be done to this particular place of that's the question. The simulation, you have to simulate each time, but to what extent are you going to end up starting
I don't know if I can answer this, what you want to hear, but our advantage is that we already simulate the observed variability in a crop. And in the case we are using stay green isolines, means that most of the features of the crops of the original background is, or genetic background is same, and we are altering only one part. No, so we are quite sure that the attained diversity or variation in the particular trait is the real one. I don't know if I understood the question properly, but I think that the key in the model is not to just change the coefficients and model something, because something may not be feasible by the crop, or some combinations of the traits which we can create in the model are not reflecting the real plant, what the real plant would look like. But in this case, we are altering just one trait at the time, and that too, the real variability which we observe. So in the case, we had our base ground simulation. In the case I was talking about those stress patterns, so that was all simulated for the common rabi sorghum genotype. So when we published this paper, then people started, who are working on sorghum across this area, they started sending emails, and they were asking how the hell did you simulate exactly what we observed here for the last 30 years. So when our base ground simulation was reflecting the reality enough, I have a strong belief that even those, when we introgress to this baseline, our trait, which is really reflecting the real variability of the crop existing in the crop, we have even very good chance that we would be very close to the reality when it comes to the real breeding. Is it what you meant? Little? Okay. Another added for coffee. For coffee with Rosana.
Okay, so outside
do you know, or anybody knows, whether that elasticity in hydraulic conductivity and the different regimes of moisture regime, is that in any way related to root anatomy?
Again, perfect question. Thank you. We are actually researching very intensively on root hydraulics, and at least we know that this trait is probably linked to the various pathways and the conductivity of various pathways, how water gets into the roots. So currently we are at the stage where we are able to separate the apoplastic pathway water flow, symplastic pathway water flow. We are even able to see and I hope that she will agree that which particular proteins, aquaporins, are probably the key for extraction of the roots. And we also know that these hydraulic properties of the cells itself can actually influence root anatomy. There are some certain indications in literature, and it is just a very initial stage, but in our millet, where we know already a lot, we know that in the genotypes which differ with root hydraulic conductivity, they also differ in the cells of Casparian band. So we don't know yet the logic, how it works, but it may be the link to this one as well, to the morphology of the root.
Basically, what we are seeing is like there are different pathways, apoplastic and symplastic. So, as you said, like at the Casparian, when the plant restricts the water at Casparian band level, so the water moves through the aquaporins. So it's a symplastic pathway which accounts for like 92% of the water pathway. So basically we are looking like how it is sensing the signals, like when the drought is there and how it is restricting. So it closes itself not to allow the water to them. And also like at some point all the aquaporins won't do that, so they don't close their pores. At some point they undergo these anatomical changes also.
So how many aquaporins are you looking at?
All.
There are a lot. Mostly PIP, I think. That's what we are trying to find out.
One, two?
It's called plasma membrane intrinsic proteins.
Yeah, which, all of them.
Not all of them. Basically, number two, PIP2. That's what we found in sorghum.
Yeah.
Like it has role actually. So
Another address for a coffee. Please come to our office and have a look at our initial results, because I think we are progressing very rapidly, especially after getting new contributions to our team. So we really know we have started with analyzing all the aquaporins at first, so that we do not miss anything. But as Josna said, really it seems that there is some key role of PIP from the two family. And we are not alone who think that. All those aquaporine guys which breed aquaporins every day, they're in France from INRA and whatever, then they actually identify the very same aquaporins, despite in different crops, would cause the same phenotype.
I agree.
So please come.
Yeah.
Any other young guys?
They are scared because I forbid them to ask anything before coming. Well, it depends how we model it. We affect only transpiration in this case. Yeah, actually, anyhow, if I lie and I say yes, then you would come to that later, but I really have to have a look at this also. This is very important. I think that with the current way how we do it, if we just impose limits flat on the canopy, we do not affect water use efficiency. In that way, which, and you know very well, that in the case we model it through the KL constant, I think we may affect the water use efficiency as well. Well, I would bet so. I don't know about this. I really don't know much. I
how
have
much
to admit.
could be different? Transpiration efficiency and water use efficiency?
Well, it depends how you define the terms. We can, once we have an agreement what is what, then we can discuss it. If we speak same language, we can. Well, you will have to penetrate the beauties of model next year, so just you wait.
I was like hoping that both will be the same, like with little difference. So only the evaporation.
But we can know only after we try.
Okay, do we have any other queries or comments? Yeah, they're being on. Thank you so much, Jana, and it's really I'm happy because most of the very good scholars are coming out really as good scientists, etc. Youths are used to get scholars,
Yes.
right? Not just a scientist with a good.
Back to the room.
So thank you so much. So we'll have let's grill more Jana. Okay. Welcome.